Thanks, Brad, for that wonderful introduction. And again, thank you to Alma for the opportunity to speak and the various funders, including Alma, that have supported this work over the years. The formal introduction will relay to you my professional credentials, and we've heard about that last night, professional credentials versus who you are. With the theme of this morning, the first speaker talking about morals and ethics, I'm actually still think of myself as a rural kid from Beaver Lodge, Grand Prairie that has a lot of family still in farming. So part of my motivation is looking at a cow or the processing of a cow like a barrel of oil. I know that's going to sound crazy, but maybe something you'd expect from Alberta. The idea here is how do we get the maximum value out of that cow? Okay, and what we've gone through in the last decade in the beef industry has not just assaulted the history of how we do things with the livestock, but it's actually directly assaulted the value that we get from that animal. And I'll go through a little bit of that with you this morning. The research platform that I'm going to talk to you today actually had a start all the way back in 2004. When I joined, my background is that I'm an industrial microbiologist, but I spent 2 years working in chemical engineering funded by Syncrude studying oil upgrading. From that, I ended up in an agriculture forestry faculty in a position you could think of as a chair supported directly by Alberta Agriculture. It was at that time food and rural development, but through all the transitions in the years. So my program has always had this applied nature, linking with government, linking partnership, and reaching across the spectra. Most of you in the room, I would hazard a guess, know this system better than I do. And what we have when we look at livestock and the cattle industry is you have this animal that goes through processing, you get the separation of the edible cuts, then you're left with the unmarketable tissues. I don't think I have a laser pointer, or I do, kind of. Okay. And basically, the whole process, if I was to oversimplify, is you take that unmarketable tissue, you break it down, you heat it to high temperature, you remove the fat content, you try and separate the bone, and you end up with the protein cuts out of that. So even back in 2003, 2004, when I was first looking at this issue, my relationship with Alberta Ag charged me to find value into agriculture. So what I did is took trips down to places like Cargill and other rendering plants and talked to the rendering industry, the processors, and said, what are the cuts that you have that are of lowest value? Or where is the opportunities to add value to the system? So even back then, we were looking at the protein cuts of what was coming out of the animal processing and saying, is there a way to get more value? Early on, and this is work we actually had funded by AVAC back then, we looked at blood meal and bone meal, and we looked at these issues and said, can we get the protein out? Can we do something more valuable with that? And our first thoughts were proteins, and it says amphiphilic. What that means is the protein molecule has a part of it that likes water and a part that doesn't like water. The combination of that in a single molecule means it wants to sit at the interface between oil and water systems. It likes to foam. It likes to have that surface activity. So we spent some time trying to figure out, can we enhance that? And we started thinking crazy ideas like, could we start making industrial products like firefighting foams? That's where this really started in the process. So we were recovering the protein at that time. It was pretty straightforward. We would then look at modification of that protein. The process, and I just want to highlight here, and there's a history here I want to draw to later, is we were looking at temperatures like 60 degrees Celsius and 4% sodium hydroxide, breaking the proteins down into smaller pieces, looking at their activity, modifying them, and really trying to get an industrial chemical out of this. And the analogy that I used earlier about the barrel of oil is, I talk about this a lot in my other venues, is that a barrel of oil, about 10% of the volume, gives the industry 30 to 40% of the value. So gasoline isn't the highest value co-product. So when you look at a cow, there's elements there that are non-food edible. Can we get more value? Is there an opportunity to make more money from the waste products than we get from the actual meat? That would be the perfect dream for me, is driving value back into the system. So this is where we were headed for a few years. We then studied things like gel chromatography, and I'm not going to go into the details here, but basically the further to the left on the graph, the bigger the molecules are, the further to the right, the smaller they are. The blue is what it looks like after an hour of hydrolysis. After 24 hours, you can see a shift. Everything's shifting to the right. That's telling you the proteins themselves are breaking into smaller pieces. So we had to get in and actually figure out chemically what we were making, what are the size of these pieces, and what can we do with them chemically. We then started working and collaborating heavily with chemical engineering. These are the expertise that the oil sand sector has been using to improve and drive their processes. And we look at things like surface tension, like how do these molecules change, how the liquids interact? Can we make better and more stable foams? And then we got into making foams, measuring them, and looking at their things like water binding ability, foam stability over time. And then we got into chemical modification. So at times we were using enzymes, but basically we started doing strange things like sticking fatty acids on the side to get more surface activity. Long story short, we were able to get, stable foams that were working really well. We're moving down that path. And then 2007 came. And this is really where the BSE crisis hit us like a freight train. At this point, we were using proteins indiscriminately from the beef industry. And what happened in 2007 is that the rules basically changed. The industry started coming to us saying, we have a problem. We have a lot of material now that we can't send anywhere. Sorry, I'll just skip through this quickly. Basically, long story short, we had a good performing form at 2007. We were doing good modifications, as I was saying. And then we had this enhanced feed ban come online. Meat and bone meal were only not for non-food, so they were reduced value. And the specified risk material was introduced into our language. This is the idea that there's elements coming from the cow that basically, for all intents and purposes, we move from that social contract you heard about into social control. Now this material, I have more requirements for handling SRM than handling radiation in the university labs. So to put that in perspective. It meant basically shutting our lab down for a year, working with CFIA, getting regulatory control in place, running it as an industrial lab under a biosafety level 2 environment, I had a research associate almost spend a year writing documentation and communicating with the stakeholders. And basically, the way I like to simplify this is, if our cow, to start the story, was a piece of pie, and that piece of pie was going to all different applications and getting value. What the BSE crisis did in my world is take a third of that pie that was selling for value, and no longer is it value in the system, it's now you're paying a penalty to get rid of it. So if you take 30% of the general industry input, and that's maybe not quite the right number, you could argue 20, 25, whatever, now charge you between $70 and $200 a ton to landfill this material into land that can't be used for agriculture. And, and that leaves you with 60% value. That 60% value, you know that processors, renders, and producers all get along really well sharing that information and dividing that value of a shrinking pie. It created conflict, obviously, within the system. Now, on top of that, you're also going to the renders and saying, by the way, can you reinvest in infrastructure to segregate this material? And can you add and spend money into the sector now that where you're making less money? That's the reality we started looking at. And so instead of, the life sciences guys look at this, and there's people here, again, know more about BSE than I do, probably, or the prion, the active component. But there's a big chunk of the cow, or anything that contacts that piece of the cow, that now has to be segregated. And this is really where the process kicked up, and APRI and Prianet and ALMA have all come together to help fund what we were doing over the years, looking at how do we take this 300,000 tons per year estimates in 2009, being in cases tipped. And I know some of the industry is looking at combustion, some of the industry is looking at maybe making fertilizer, but I don't want to just repair that third of the pie. It would be nice if we could take that third, put it back, and increase it to half the value of the pie. Maybe I'm dreaming in Technicolor. But it does, when CFI comes forward and says you either have to landfill it, which is cost, you can incinerate, well, this stuff has a lot of sulfur in it, a lot of nitrogen. It's not the best stuff to burn, but you can get some energy from that. Or we can use thermal and alkaline hydrolysis. So we can get aggressive breaking this down to make it safe, but that comes at a high cost. So you need to have a high value back end, in my mind, to justify that process. So when we just compare some of the options, we can landfill it, we can make energy, we can make feed, fertilizer. Functional utilization is where I was really wanting to go. I want to take that protein, render it safe, and now make something more valuable for it. What we started looking at things is bioplastics, biocomposites, adhesive flocculants, and surfactants. This is where we'll go. So our initial projects coming out of that kind of restructuring of our program were to look at high-value industrial applications. So we're not looking at making food packaging out of this stuff. We could have it completely safe, but no consumer is going to want to have something that was a risk material now associated with their food. So we start looking at high-value industrial space. So this is where this is foreshadowing plastics, composites. We started thinking we need a large volume application. When we're talking to our partners like Sanimax or West Coast Reduction, they're coming to us saying this is a lot of material. We don't want to make a 0.01% component of a surfactant that we sell a few tons a year. They want to move huge volumes. So we're looking large things like, they talked a bit about insulation, packaging. Even if we could make fence posts that go into farmer land, there's kind of a continuation there. Or car parts. So what we had to do is go back and redesign our process in the lab. We're at about 1 liter scale here, studying both the alkaline and hydrolysis and thermal hydrolysis. So the question here is, if we use a caustic base to break this stuff down, what do we get? Nobody in literature knew what you're getting. They knew it was safe from prion standpoint, but nobody understood the nature of what we had. Were we going to have amino acids, small proteins, or what? Likewise, we looked at the thermal process, which is, again, higher temperature. The thing to identify on this graph is now we're talking 150 degrees Celsius. This is a little different than the 60 degrees and 4% caustic, okay, where we're now 15% sodium hydroxide. This is the stuff, and I don't want to be graphic, but you see the CSI movies and people are dissolving bodies away down bathtubs. We're in that domain, really aggressive, okay? Sorry. The idea is, after we characterize the sizes of what these little peptides, and it actually did form small little pieces between about 3, for those of the science in the audience, 3 kilodaltons to 10 kilodaltons, so about a 10-fold reduction in size of the protein from what was there. We then started going, as a polymer, we engaged guys like Philip Choi, Tony Young in chemical engineering that are polymer scientists. We started looking at traditional cross-linking chemicals that would come in, and we really are trying to form a matrix network out of those little pieces. So we took big proteins, broke them down into little pieces, and now we're trying to synthesize them back into a network or a solid material. So we looked at everything that's out there, wide range, glutaraldehyde, benzaldehyde, epoxy resins. We kind of went across the board and screened everything. And actually, out of that, we started filing some patents. We actually were able to make plastic material. I actually have, if anybody's curious, I have samples of everything I'm going to be talking about today in my bag. I just didn't get them out. So if you want to see them, I'm happy to share them. The next thing, the science level. So we didn't approach this like bucket chemistry. We went back and on a model compound basis started studying things. Well, these proteins, these little pieces still have nitrogen on one end. That's part of being a peptide. So what you could do is if you had 1,000 pieces of molecules, you have 1,000 nitrogen on the one end sticking out. What we then is went back and we're monitoring how much of that nitrogen could we detect. So if you look on the far, under each of these cross-linking reagents, glutaraldehyde or resorcinol, on the far left is an example of something with no cross-linking. That's how much nitrogen we were measuring. And what you'd see is in every case, the nitrogen that's free and accessible is disappearing. That's our indication that the nitrogen is being engaged and being used in that cross-linking reaction. So basically, the more that goes down, the more we know we're getting cross-linking, the more we're tying up that protein into our solid material. Then what we did is we started getting, this is where we had a bioplastic. So this is about 2 years ago we're at this point. We started then characterizing what this plastic looks like. So there's a lot of performance criteria if you're going to move into the industrial space that become important. And so there's everything from looking at the chemical reactive groups on the protein that we could see. That would be the FTIR. We look at stress strains, so mechanically, how is this plastic behaving for us? Is it brittle? Is it pliable? We then looked at things like DSC and TGA. These are methods that are really studying the chemistry and the energy of those reactions. So but basically, in one case, you're heating it up, and you see the energy being taken in by the system, and at some point, the reaction takes off, and you see a big change in the energy happening within that system. So from those approaches, we did things like FTIR, where we could see, again, the top blue is our protein that's been hydrolyzed. What you see here is every one of those little peaks that comes down is the indication of a chemical group. Maybe it's a hydroxyl group or whatever. I'm not going to go into a lot of detail. But you see a characteristic signature of our protein. The green lines are the epoxy resin, so chemically that's kind of showing us what groups are there. And what you see here is when you bring the two together and form the polymer, we get massive changes in what peaks are there or not there. So we're actually seeing and now confirming we're getting chemical reactions between the two components. From that, we then started asking other questions like, how will our plastic behave in water? So what you do is you take a piece of your plastic and you put it in water for 24 hours, submerge it, bring it back out, and then you measure things like how much water did it take up. Then you dry it down to get rid of the water and see how much of the plastic disappeared into the water when you had it soaking. So you look at, and for any plastic, you want to have minimum water absorption, and you don't want to be losing your plastic into the water as it's dissolving away. So this is the kind of studies that we were doing. And we were getting a very high water, and I'm kind of bouncing between the different cross-linkers that we're using just to give you examples. But this is the epoxy, for example, we were using. It's quite well performing. In other cases, we were comparing that thermally hydrolyzed material. So this is just high temperature breaking the protein versus using caustics. And the problem with using caustics is you have to neutralize it. If you have to neutralize it, you're making salt. So we can start looking at things like accelerants now. Can we add something to bring that energy down and make a better, more economical process? what you start making is different types of plastics. And I have a grad student that likes to dye things, so you know cow parts don't become orange or green. But this is the idea. If we start generating materials, we wanted to look at that. And those little dog bones, as we call them, are what are used in that stretching experiment I talked about, the mechanical testing. So we cut those out, and I have some of them here if somebody wants to actually touch, feel what these look like. Just the protein, the resins tend to make fairly rigid structural material for us, so it's fairly brittle. So what we were able to do is that we got our cross-linking reagents, et cetera, to work fairly well. Epoxy tended to be the best performer from our criteria, so we've really focused on that. And we started now reaching out and talking to the chemicals industry. So we started talking to some of the people, and I can't go into company naming, but are already studying thermal hydrolysis, and there's discussions with them. We're going through the process of patenting this. We're talking to companies like Tessenderlo, that are a Belgian chemical company. It's about a $2 billion company, works in things like animal byproducts already. So they're doing things like collagen and that industry, and they're very interested if there's linkages that can be done with their industrial chemicals division. So we're getting those materials very characterized. We've got up to 5 unique cross-linkers that give us different materials and different properties. And we're really focusing, though, right at this point on the epoxy. So that kind of takes us through to about 2 years ago. Then we started saying, well, plastics are nice, but can we work with other partners in the Alberta Innovation System? And in Alberta, we're very fortunate to have a group called the Alberta Biomaterial Development Center, which is a linkage between the old ARC, which is now Alberta Innovates Tech Futures, and Alberta Agriculture, that has basically up to 1 ton per day level activities in fiber from renewable sources. So we started thinking, well, let's look at composites. So what is a composite? It's basically the idea that you're taking some kind of rigid support and then embedding that in a resin or a plastic. It's almost that simple. So we bring a matrix, which is the plastic that we talked about, some kind of reinforcement. And the best example everybody here would know about is something like fiberglass. So you've got glass fibers encased in a resin. In that case, it's completely petroleum-based or silica-based, non-renewable. The province is investing a lot through AI Bio and others, looking at can we take hemp or wheat straw, pull the fibers out of that and use that for composites, maybe with non-renewable resin, or can we, in our case, form a resin or a plastic that can go around that? So as I was saying, you have your filler, which is your fiber, your matrix holds the whole thing together, and really you get a high-performance material that's better than either the plastic and the fiber itself isn't the material, so you're really embedding it. Where you're seeing examples right now that are exciting and very high value is you start hearing things about Boeing or Lockheed Martin making these advanced planes that can fly twice as far. They're really going to these kind of advanced composite materials to give them the structural strength at much lighter weight than using reinforced steel, for example. So that's kind of where we start. As you're noticing a tone, we're talking higher and higher value and trying to push the behavior of this material. And really this is, again, where Alma came in and completely was driving this with partners, is that we want to look at building these higher strength, higher value material. We're confident our plastic is going to perform, but if we're going to do something and go commercial, let's get the highest value right out of the gate. And this is just reinforcing, again, same kind of thing, different words, but historically you would use in a composite. Another example people would know is something like concrete or an aggregate. You've got the smaller components and then you're embedded in a bigger system. Now the advantages, if we get to the renewable fiber, the renewable materials, we're going to have environmental benefits, we believe, lower cost, lighter weight, lower density, and they just have better performance criteria if you design them properly. And one example, another nice Alberta example that's online and linked to the ABDC and supporting it is the Kestrel coming out of Southern Alberta, Motive Industries. And again, this is a car built with this material. So this isn't an esoteric academic exercise in the university putting strange things together. The industry is actively trying to find these materials that will perform better. And if you do it right, it will work. And there are examples online right now. So just to bring that kind of to a reality stake. From the biocomposites, our research side, again, is to develop this material. How do we introduce the cross-linker? And now we're twisting our chemistries a little bit because we don't want to just make a plastic. We want to make a plastic that's going to interact with the fiber. So it's a little bit different chemistry we need to go back and study. And also we start to ask questions about, well, the fiber we're talking about, do we need to modify the fiber so that it's more compatible with our resin? And so we developed these systems, and this is where the program really started working a lot more with AITF, and guys like John Wolodko, Trevor Click, again, from Alberta Ag is really key to bringing a lot of these teams together. And so we started looking at everything from industrial hemp and flax, and wanting to bring that into the overall system, working with Vegreville. And they basically provided us fibers. We started looking at bringing them together, and we've just gone through a whole suite of testing. And this is where, as I keep highlighting them, but it's so wonderful from my standpoint to have all these different ways of putting the composites together. So we can lay the fiber down, press it, we can weave the fibers into a mat, put the plastics around it, we can inject mold. There's a lot of ways we can do that, which makes us all a little bit different types of material that we can then go into testing. And again, our problems, as I highlighted, is getting our resin to interact with these fibers. Traditional composites, you have things like glass and petroleum-based resins, which interface fairly nicely. What we're talking about is moving into a world where things are more water-loving, a little bit more polar, and you're trying to bring that association together. And so we're trying to make our matrix, in our case, we're trying to make the matrix better so it interacts better with those renewable fibers. And again, doing a lot of the material property characterization, making these press mats, and they make nice little plates with fiber wound. It looks exactly like fiberglass, except that it's different colors and it's fiber, renewable fiber. And then we do things like take those and stick them in the soil and study over a year the biodegradation. We ask that question, look at water binding, look at temperature stability, and that kind of stuff. So out of that, then we do go back and everything I talked about with the plastics. We have to do the water uptake, we have to do stress strain, we have to go through the whole thermal, like at what temperatures are things reacting, how do we get the best adhesion, do we still have reactive groups, are we completing? All those questions are right there, but now we're also moving into the biodegradation and moisture resistance. And just to give you an example here, again, we're looking at the epoxy, modifying this resident, and you can see, and you probably don't remember, but we're driving the water binding down. The water absorption is now like over 1%, which is about 3 times better than it was earlier on in the process, and the weight loss. So the idea here is we have to continue the research on making the resin better and the chemistries to see how it interfaces with the material. And so where we ended up with that is we basically are in the process now of finalizing some final testing with that, and that was about a year's funding that brought us up to the beginning of this year with ALMA support. And then where we take it from there is where we are now, which is now that we know that we can make plastics, we can make these biocomposites, the next element that we looked at, and this is actually kind of interplay with ALMA, it was great to have that resource of the forestry community that we're also touching with to say, can we look at adhesives? So this is another reaction of those pieces in some more chemistry. Can we bind things like, can we make plywood? That's a simple question. Can we go in and replace some rather nasty crosslinkers that are being used with these very renewable kind of polymer systems? The other one is the wood pellet industry. So we're talking a lot to the Wood Pellet Association and BC, research centers at UBC. Can we take our material and use it to make wood pellets? Now this is another application. We don't want to make it too strong. I mean, we want to have these pellets that you can stack them, 50 feet high and not have the bottom ones exploding from the pressure. But at the same time, they have to be able to put them into a grinding mill later for combustion. So we got to get just the right performance criteria, and that's really, over the last 3 months, we've really launched on this project. And it's nice if we can remove things like formaldehyde and go to a cheap, abundant application. So we've formulated different systems on our SRM protein hydrolysates, and basically looked at things like oak veneered plywood. So you basically do studies where you put a layer of our resin, put the woods together, let it anneal, and then you do all that structural strength and adhesion and water binding and all the rest. And you do criteria like how hot can you run the system before the wood starts decomposing? At what pressure now do we do this reaction before the wood starts crushing? Is there an optimum? And we're doing a lot of process optimization there. And again, you put these plates together and then you pull them apart, check your shear strength. Again, I have some samples. And so really, this is only 3 months in, but we're already getting a lot of these composites and it's performing really well. I'm actually really excited. Again, I have samples if somebody wants to play with them afterwards, but we're working towards further improvement. So in a nutshell, what we're really trying to do is, I'll go back to the analogy. How do we take that third of the pie that disappeared? How do we put it back in the system? How do we grow the pie? I know a lot of what ALMA and the food industry is doing is looking at the other 2/3 and trying to grow that value as much as we can, and that's great. But I want to focus, at least our lab, on that last third and driving that value back for everybody. So with that, I mean, I have a co-investigator, Philip Choi from Chemical Materials Engineering, kind of the true polymer scientist. I'm more the reaction guy. And then collaborators across the system, AITF, Woodbridge Foams has come in as a big car part manufacturer. They're one of the large Canadian firms that sell car parts across the world. Some discussions with some other partners like Magda, et cetera, in the background. We're even talking to enzyme companies like Novozyme on bringing these systems together as well. So with that, and I want to highlight, everything I'm talking about isn't me, you probably guessed that. It's been a continual team of usually 2 grad students and a postdoc. And so everything you're seeing brought together is that interdisciplinary play between materials engineering, chemical engineering, and the life sciences. So with that, a wide board of support we've gotten, especially Alma, AI Bio, the Prianet, APRI, and across the board with industry. So with that, I'll conclude, and thank you for your time and attention if there's any questions.